So as you would imagine with exercise, and in this case resistance exercise, you do burn through a lot of energy, or like you use a lot of energy just to reduce those muscle contractions. So a priori, mitochondrial health or mitochondrial characteristics could be an important factor to ensure that enough energy is produced within the cell.
Hello, Javier. It's my pleasure to have you in Evidence Strong Show. If you could briefly introduce yourself.
Hello, Alex. Thanks for having me. My name is Javier Botella, and I'm a postdoctoral researcher in the field of metabolism. In the past, I've worked specifically in muscle physiology, and then moved a little bit into cardiac metabolism and more the health area. And I'm now working in a little bit of a different field in immunology, trying to explore new areas of research.
Awesome. So I invited you to pick your brain on the topic of muscles, specifically muscles of strength athletes and how they possibly differ from untrained individual muscles. So the paper we'll be talking about is titled Increased Mitochondrial Surface Area and Cristae Density in the Skeletal Muscles of Strength Athletes. So why this study?
So this is a collaboration that we started after I submitted my PhD, and we were in the COVID times in Melbourne. I had a lot of free time, and they had done, this group in Denmark, they had done a prior study on looking at the glycogen depletion. So the glycogen stored, the energy source of one of the energy sources of muscle contraction, how it was depleted in elite powerlifters and Olympic weightlifters during an exercise session. And when I was reading this paper, I remember seeing some of the electron microscopy images. So these images have like a really high resolution, and you can see the contractile fibers, you can see the glycogen dose, you can see the mitochondria as well very nicely. And I remember seeing one of these images where I could see a single mitochondria that was slightly, in my opinion, damaged, and that really caught my attention. So then I talked to these researchers and said, have you thought of looking at what happens to mitochondria in these samples? And then this was a bit of a beginning to see what was going on, because I think until then people thought that strength exercise or strength trained people had a decreased mitochondrial volume, and it was kind of not detrimental, but it wasn't benefiting mitochondria of these people. So then we started this analysis, and then some of the results that came out that I will explain later were really interesting, and then we kind of developed the research project and the idea as we were getting new and more interesting data.
That's an awesome story. So it's kind of not an accident, but that's your curiosity that led you to start the study.
Yeah. No, it was really curiosity, and also like the right time of having a bit of a free time as well, and then the openness of other researchers from the other side of the world to share their valuable data and so on, to go further and explore a new area of research, and this was the case. Yeah, it was, and I think the outcome has been, I'm very proud of it. It's like a very nice story as well, and I think it's maybe I'm being too optimistic in this regards, but I think it's going to change a little bit the dogma traditionally within mitochondria and strength exercise, or I hope so at least.
I agree. So before we go into what have you found, we have to know a little bit more about what the study was about. So you hinted us a little bit, the samples were taken in Denmark. Could you tell us a little bit more about the process? Because not everyone is familiar with the procedures. How do you take the steps and so on?
Yeah, and I'm also now thinking that maybe not everyone knows what mitochondria are exactly, so I can also go through some of it as well.
Awesome.
So these studies are usually invasive studies. So I did it also during my PhD. In this study, I didn't do it myself, but like they did it in Denmark, work with medical doctors. And then what they do is you anesthetize a specific region of the vastus lateralis, which is the more external muscle in our quadriceps. And then you make a small incision, and then you go with a needle, and then you make like a little chop on the muscle. And then through suction, then you bring it out of the muscle. And then that's your piece of muscle, which is usually the size of a few rice grains. So it's around 100 milligrams. And then with that muscle, you have a lot of information that you can work from it. And is it a little piece that will go frozen, and then you can do gene expression levels, protein expression levels, and a bunch of things. For the electron microscopy specifically, because we want to preserve the whole structure and the morphology of that muscle, it's fixed into a specific chemical. So it's straight after bringing it out, you fix it, and that preserves of the whole morphology, and then you take it through a series of processes of as any protocol, and then you do the high magnification imaging. So this is the biochemical part, and then these studies, what they've done is that you bring in the patient, the participants, you give them a standardized meal in the morning. You try to standardize as many things as you can with your participants just to avoid any co-founder effects. Then you do the initial biopsy at rest. So that's your resting point. Then you do the exercise study or the exercise intervention or whatever it is. In this case, it was 3 exercises. It was squats, and then it was deadlifts, and then it was split squats as well. It was 4 sets of 5 repetitions at around 70% of 1 repetition maximum for the 3 exercises. And then straight after you get the last biopsy, and then you can compare across and with individuals. Then the other thing I didn't say, it's like what mitochondria are. So mitochondria are these organelles that are very fascinating for those that work on it or like know a little bit about it, because through evolution, we initially didn't have them. So this is the theory comes from bacteria. And then at some stage they were incorporated into eukaryotic cells, and then they worked together to kind of help each other. And what mitochondria provide is nutrients to the cell, and that's by means of using oxygen. So it's basically providing the ATP, the energy sources for the muscle to contract, or at least the most efficient type of energy that can be provided to a muscle cell or any type of cell. So as you would imagine with exercise, and in this case resistance exercise, you do burn through a lot of energy, or like you use a lot of energy just to reduce those muscle contractions. So a priori mitochondrial health or mitochondrial characteristics could be an important factor to ensure that enough energy is produced within the cell.
Awesome. Could you say a little bit more about the participants who you recruited for the study?
Yes. So it was 10 male participants. The age was around 24 years old, and they were all local within Denmark. Consider elite athletes or elite strength athletes. They were both powerlifters and Olympic weightlifters. They were well-trained athletes, and this is, I think, one of the beautiful things of this study, which is not really easy to get very well-trained people sometimes.
We know who participated in the study. The samples were taken, then processed so that you can see them under the microscope and take some photos. And what happened from there?
Yeah, so so they take a bunch of photos. So usually they select around 6 different fibers. Through the images you can estimate or determine, depending who you ask, whether it's a type 1 or type 2 fiber. So then this was all done in Denmark, and then this also ensured that we were blinded throughout the procedure. And then they identified 6 fibers, roughly 4 to 6 fibers of type 1, of type 2. And then they took about 6 to 12 images of different sections of the fibers. So they took those images. So a lot of images, then barcoded it with different IDs. And then that image is sent to another researcher with numbers. And then everything is analyzed through a software called Image J, where all you do is when you see a mitochondrion, you just circle it and then you add it to the list. And then you do that through images and images and images. I think in this study, I quantified or I circled around 30,000 mitochondria, something like that. So it's like a lot of work in there. And then what you do is you use any statistical software or similar, and then you create these curves of distribution of the mitochondrial size, of the morphology. And some of the findings that we saw is that the morphology was quite different compared to a normal muscle, even though the amount of mitochondria was relatively similar across participants, untrained and trained. And we can go into this detail afterwards, but that morphological assessment was really, really key in this specific study.
So we know that mitochondria are these organelles that are in the muscle. They are responsible for, let's make it simple, for producing energy for the muscle to work. And now when we go inside or we cut it and look into it, how do they look like?
Yeah. So why is mitochondrial morphology important? I think that's a good question. And so there's a few ideas in the literature, or like evidence in the literature, and one of them is that the mitochondria within a muscle forms a reticulum. So it's like we take 2-dimensional images, so we see like a circle here of a mitochondrion, but we don't know in a 3-dimension whether it's it's a tube, or whether it's like a pipe or something, or whether it's a very isolated one. Some of the evidence in the last 10 years suggests that even the mitochondria that are in the sarcolemma, they are linked to the ones inside, and it just forms like this network of organelles. They all work together as a shared kind of entity somehow. However, there's other people that think that they are better if they are isolated but just next to each other. So if something goes wrong, you can get rid of one and then the rest are fine, because if you have let's say a network, then they can collapse together. And this goes very match with the environment they live in, right? So if the morphology is important because if they're larger, maybe they're able to distribute energy better, but they can collapse easier. It's hard to know. If they're isolated, maybe they can also handle easier their environment within that area. I know this is a bit of a complicated, and I think it's because it's not fully clear also what's good and what's bad. But mainly, let's try to simplify it. What we think is like a small mitochondrial morphology. They have more surface to interact with their environment compared to a bigger one per volume. So this is another tricky thing, per volume. So what might be happening is that these mitochondria are actually more fitted to, let's say, confront the calcium flux that happen with contraction, the energy, alterations that happen with exercise, and so on. So it's just being, let's say, more prepared for their size compared to a bigger one. So one of the different characteristics that we saw within the strength training participants that had also been reported with the endurance trained individuals is that they had increased cristae density. And what is cristae? It's this. So mitochondria have two membranes. So it's the outer membrane, which is the one that gives you the size of the mitochondria, and then the inner one. And the inner one is where the electron transport chain or oxphase system is found. And this system is the one that produces the energy, the ATP. So by folding this membrane more times, so creating a higher density, what you're actually is maximizing the efficiency of a single mitochondria. And that's what we found, that was roughly, I think, almost 20% increased in resistance strain. So they had 20% increase of that inner membrane compared to the volume of the outer membrane. So they are roughly 20% more efficient or more effective at producing ATP.
Wow, that's a huge number.
Yeah. No, it's big, and this is very important, and this probably has an evolutionary pressure in the sense that skeletal muscle, a strength athlete wants to have increased force. You need those myofibrils, but at the same time you have a limited space for everything else, right? So pretty much putting under pressure the whole time mitochondria, and then the only way they can adapt is really by maximizing what's inside them. So it's a really impressive adaptation, I would say.
So you strength athlete make the mitochondria more efficient, more denser, but not bigger.
Yes. So I think they don't make it bigger because they compete with other processes. So because you need more than probably endurance athletes, the muscle contractile proteins. So you need those for the muscle contraction. And that's probably the priority. So the amount of space that's left within the muscle, it's really tiny for other organelles and so on. So that's the way probably mitochondria has adapted through resistance trained individuals. It's like, okay, we don't have enough space. What can we do to be better? It's like, okay, so we need to improve our cristae density, and that's what's happened.
How fast does it happen?
So this is a very, very, very slow process. There's a few studies that have been done trying to look at whether it changes with training, and so far no one has seen it with training. Of course, there's the argument of whether it's something that's in there on the strength athletes that makes them athletes, or whether it's over time generated. I think it's over time, but I think it's over years that you need, because it's a very impressive adaptation from the biology perspective. So I think you need to push those mitochondria like day in, day out, and then they slowly adapt in that way. So yeah, it doesn't happen overnight. It doesn't happen over a month or a year. I think it would happen over years at least.
Makes sense. The process of development of strength athletes takes years. So I can see how what you're saying agrees with what we observe as coaches or as athletes. Do you have any idea on whether this adaptation could have negative effects on health or other aspects?
Not at all. I think if you look at the only aspect I could see as a negative effect, but I still don't think it's a negative effect. But if you read the literature, you might think it's a negative, is that the volume, the amount of mitochondria that you have, because you have a limited space in the muscle, it's low as a percentage. Mm-hmm. But then if you think that strength athletes have a higher muscle volume, at the end of the day, they actually probably have more mitochondria as an absolute value than normal people. So I don't think at all this is a negative effect. I would say the opposite. It's a very positive effect.
Okay. Do you have anything to say about gene expression?
Yeah. So we saw this enhanced cristae density structure. We saw that morphology was slightly different. So the mitochondria were relatively smaller compared to the untrained people, and they had like more surface to interact with their environment. And what we saw is like following a single session of the exercise, strength exercise, this morphology got into more isolated and probably a little bit of a stress effect on this mitochondria. So then we followed this with the RNA sequencing and RNA experiments. And what we saw is that genes related to stress responses, specifically mitochondrial stress responses, were actually activated during following strength exercise. And this has been shown, and we have some work ongoing that this has been shown also in high intensity endurance exercise, so some of the beneficial effects of this type of endurance exercise. So we confirmed that it also happens with strength in a trained cohort. And then what we saw is that when we compared the transcriptome, which is like the baseline level of gene expression in a trained cohort and an untrained cohort, we also saw that those genes are generally increased or augmented compared to the untrained individual. And this mainly means that they're more prepared to cope with homeostatic disturbances, with muscle stress, so that they're very prepared for it.
All right, so strength athletes, because of the training, their genetic material is prepared to handle that height of training.
Yeah, I mean, it makes a lot of sense because they are exposed to it very often, so they just have the coping mechanisms to recover properly. So yeah, you're right.
That's awesome. And would you have any advice to coaches and strength athletes how to use this data?
I mean, this data by itself, it's a bit of a more like basic research in that regard. So it's hard to really make an advice or a tip to coaches or scientists in the area. And I'm definitely not the expert in that area. What I would say is like, there's one more factor now to take into account probably within this field. This is like the mitochondrial health might be key, or it's a contributing factor to the performance of your athletes. So just make sure that keep up with the literature in the sense of, regarding mitochondrial health, what other things you can do to enhance the recovery of these specific, organelles and so on. And I think at the moment, there's not a lot of evidence to really give a tip or an advice, but probably in the coming years, it's becoming a very interesting area and an ongoing area. If there's things you can do to enhance the recovery of this mitochondria or the recycling of this mitochondria, it might be an interesting approach for your athletes as well.
Ooh, something to wait for. Do you remember how long the athletes were training for? What was the experience for the athletes in the study?
Yeah, so they had an average of 7 years of experience.
All right.
Yeah.
Okay, 7 is a good number. Yeah.
So I think coaches definitely know this. It takes a while to get those adaptations, but it's good to know that nature is doing a good work in sharing that over time you get those adaptations.
Okay, so we still have some questions. We will have to wait for new results to come. So just to wrap up, 2 short questions. One is, what is your favorite color?
My favorite color is red.
Awesome. Noted. The last question is, if someone wants to contact you online or keep up with your work, where they can find you.
So at this stage, you can follow me on Twitter. Every now and then I retweet papers or like something that I feel is interesting, or on LinkedIn as well. You can follow me but it's more about my profile, where my career takes me. So I would say Twitter is more of an interesting way of following my work and also some interesting research that I share.
Is it your name? Your handle is your name?
It's Javi. So maybe you can write it afterwards in the podcast or something, but it's Javi_IUP. So Yup, it's a name I picked way before I was a scientist. So I wouldn't have taken that place.
Yeah, now it will follow you.
Yeah, now I cannot change it. So
awesome.
it's fine.
Thank you so much, Javier. It was my pleasure.
Yeah, thanks a lot.